I thought I'll give a brief introduction about the EMT and how we got there, and as well as its relation to cancer progression or cancer biology. So we know that more than 80% of the cancer which occurs in human beings are epithelial in nature, or carcinoma. Now, when we also know that we have good therapy treatment option for carcinoma, but most of the patients, they give up their life because of metastasis or tumor resistance or tumor relapse. Now, if you look at a metastatic cascade, you have a primary tumor and then become highly angiogenic, which eventually some tumor cells from the primary tumor enter into the circulation. They travel in the circulation, exit the circulation, and some tumor cells find a way to form a micrometastatic colony, and those tumor cells eventually develop into secondary tumor, which is again highly vascularized, and that seeps into other organs, and then the process continues. Now, if you just look at a normal organ architecture, you could see, for example, just looking at colon, and the cells are beautifully organized, and they are attached with one another through a number of cell adhesion molecules, such as adenine junction, gap junction, desmosome, hemidesmosomes, and etc. Now, if a cell like this gets transformed, we form a tumor, and most of the time they remain as a benign tumor. The reason is they still continue to express these cell adhesion molecules, which we just discussed. So, if you look overall how these epithelial tissues, which forms more than 80% of the cancer in human being, how they are organized, they are attached with one another through cell adhesion molecules, and they, such as cell, and also the tight junction, desmosomes, and gap junction, and there's a cell matrix interaction through hemidesmosomes, and and also these cells are beautifully well organized through a number of cytoskeletal filaments, and they are all polarized. They are not simply sitting like the way we grow in culture. They are polarized in an organ setting. So now a cell like that gets transformed. If it has to complete all this step, it has to break this cell adhesion boundary and then enter into the circulation. So again, it's important that the cell has to lose cell adhesion, cell matrix interaction, gain some kind of cytoskeletal changes, and lose polarity, and then those cells can get into the circulation and travel to a distant site and establish a secondary tumor. So a while ago, when I was in Bob Weinberg's lab, there's the two postdocs at that time. Both are William Hahn is a faculty in Harvard, and Brian is working for a company. They took normal human mammary epithelial cells, and they introduced a set of genes such as S-reporting early region, telomerase, and oncogenic form of RAS. When they introduced these genes into these normal human mammary epithelial cells, they could transform these cells. Now, when they put these cells into the mice, they found a tumor, but the interesting thing was they found non-metastatic tumor. So I asked, could I take these cells and introduce additional genes and make them more metastatic? So that, particularly for spontaneous metastasis. So what is spontaneous metastasis? If you look at paper, most of the metastasis studies are done by introducing cells